All right, today we're diving deep inside the living cell. But here's the twist. We're not putting on our biologist hats. Nope, we're putting on our engineering hard hats. Why? Because we're going to talk about reprogramming the fundamental machinery of life itself. No, I'm serious. Just think about that for a second. What if you could program a living cell just like you'd program a circuit board? What if we could look at a simple yeast cell, not as some big biological mystery, but as a tiny programmable gadget? Because if you can do that, the stakes are, well, they're massive. We're talking about turning these microscopic factories into powerhouses that could solve some of our biggest problems. I mean, manufacturing clean energy, creating life-saving drugs, building a more sustainable world. And this isn't some far-off sci-fi dream. This is happening right now.
And the name for this whole field, this big idea? It's called metabolic engineering. And really, it's a total shift in how we think about biology. We're moving away from just observing life to, actively redesigning it.
Okay, so if you want to start programming a factory, what's the first thing you need? You need the blueprints, right? The schematics. And for the cell, that's where this new way of thinking called systems biology just completely changed the game. See, for the longest time, we kind of pictured the inside of a cell as this chaotic soup of chemicals bumping into each other. But systems biology gave us a new pair of glasses. It let us see that it's not chaos at all. It's an elegant, logical network. Think of it like a city's power grid, or even better, a super complex circuit board. And the scale here is just mind-blowing. In a single human cell, you're looking at more than 860 of these unique metabolic pathways. And they're all running at the same time, all connected to each other. It's wild. You can't possibly understand it by looking at one little piece. You have to see the whole system at once. So to really wrap our heads around this, let's stick with that computer analogy. Let's think of our body's metabolism as a giant motherboard. The brain, that's your CPU, always demanding power. The liver, that's the power supply unit, the PSU, controlling where the juice goes. And then you have these key molecules and signals acting just like USB ports, logic gates, and all the buses that carry information. This analogy, it's everything. It's the key to understanding how we can actually engineer this stuff.
Okay, so if the cell is the hardware, the circuit board, what's running the software? What's the operating system? How is this whole factory actually controlled? Well, it turns out it's not random at all. There are very clear rules, a real chain of command that governs the whole operation. All right, so all the traffic inside this cellular city flows through these critical intersections called metabolic hubs. And the grand central station of them all is a molecule called acetyl-CoA. Seriously, think of it like a universal adapter or a USB port. It doesn't matter what you eat, carbs, fats, proteins, your body breaks it all down into this one single molecule before it can be turned into energy. So if you can control this one hub, you basically control the entire fuel supply of the cell.
And managing this whole system happens on two different levels. First, you've got this super fast local control called allosteric regulation. This is like a logic gate right on the chip. It's an instant feedback loop. If an assembly line starts making too much of something, that product literally goes back and physically blocks the machine that made it. Bam, instant shutdown. Then you've got the slower, bigger picture control, hormones. This is like the system management bus, sending commands to the entire organism, telling everyone whether it's time to store energy or time to burn it. And really, that whole system-wide command boils down to a battle between two main hormones. Think of it this way. After a big meal, insulin is the boss. It runs around telling every cell, hey, we've got supplies, store this energy, pack it away. But when you're fasting, a different hormone, glucagon, takes the stage. Its job is to say, all right, everyone, release the reserves, the brain is getting hungry. And what's so incredible is how perfectly coordinated this all is. When you're in that fasted state, it's not chaos, it's a symphony. The liver acts as the giver, releasing glucose into your blood. Your adipose tissue is the supplier, sending out fatty acids for fuel. And your muscles are the ultimate adapters. They cleverly switch to burning that fat, which saves all the precious glucose for the one organ that absolutely needs it, your brain. It's just a beautiful, efficient system. Okay, so let's recap. We've got the blueprint, we understand the operating system, so now for the really cool part, hacking it. This is where metabolic engineering stops being about just watching and starts being about doing. And the goal here is, well, it's simple, but it's also incredibly audacious. See, a cell's natural programming is all about one thing, its own survival, make more of itself. Our goal is to go in, rewrite that code, and basically tell it, look, forget about all that, you've got a new job. Your primary function is now to become a tiny factory and pump out this one specific product that we want. So how do they actually do it? Let's go back to that circuit board. If a pathway, a signal, is too weak, you just boost it. You can tell the cell to overexpress certain enzymes. What if energy is going down a path you don't want? Simple. You cut the wire. You just knock out the gene for that competing pathway. And maybe the most amazing part, you can install a completely new circuit board. You can literally take genes from a plant, for example, and plug them into a yeast cell to give it a brand new capability it never had before. And this isn't just random tinkering, trial and error. It's a super rigorous engineering cycle. Design, build, test, and learn. First, you use powerful computer models to design the changes you want to make. Then you go into the lab and build it, using genetic tools to edit the DNA. Then you test it. Did it work? How much product are we getting? And finally, you learn from that data to go back and make your next design even better. It's this constant loop of improvement, getting more and more efficient with every cycle. Now, for decades, that build step was the big bottleneck. It was painfully slow, it was clumsy, it was super expensive. But then, a revolutionary tool came along that just changed everything. You've probably heard of it, CRISPR-Cas9. It's basically a molecular scalpel, a search and replace function for DNA that gives scientists the power to edit genes with incredible speed and precision. And the impact of CRISPR, you can't overstate it. It was a revolution. Things that used to take months or even years of painstaking work can now be done in just a few weeks. What used to be expensive and inefficient is now cheap and accurate. And instead of editing just one gene at a time, scientists can make complex, multi-gene edits all in one go. CRISPR basically took metabolic engineering from being kind of an art form, a lot of trial and error, and turned into a true programmable science. So what does all this mean? What does this programmable future actually look like? What are these little cellular factories making for us today? Well, the results are already here and they are already changing our world. Let's start with medicine, where the impact is just incredible. Take a drug called artemisinin. It's a critical anti-malarial drug, but it used to be really hard and expensive to get. Well, scientists engineered simple baker's yeast to produce it. The result? The cost plummeted, the supply is stable, and it's saved countless lives. And we're seeing the same thing with things like next-generation insulin and even new compounds to fight cancer. This tech is also at the absolute heart of the green revolution. Instead of drilling for oil, what if we could just grow our fuel? We can now program microbes to turn sugar into advanced biofuels like butanol. We can engineer algae to become little biodiesel factories. We're even creating bacteria that can produce high-quality biodegradable plastics from scratch. This is how we can start to build a world that doesn't rely on fossil fuels. And yet, it's even changing what we eat. Are you worried about a future without chocolate? Well, don't be, because scientists have engineered yeast that can produce a substitute for cocoa butter, fixing a really fragile supply chain. Beyond that, we're using microbes to create essential vitamins, natural flavor compounds, and even engineering plants themselves to be more nutritious. So the big takeaway here, the crucial point, is this. We've turned a corner. We have fundamentally moved from simply observing life to rationally programming it. Biology is no longer just a science of discovery, it's become a science of design and creation. We have the tools right now to program living things to solve our biggest problems. And that leaves us with one final, and maybe the most important question. We've established that life's code is editable. The cell is a programmable factory. So really, the only limit now is our own imagination. With a tool this powerful, what problems should we solve next?